Welcome to my channel. Now we are discussing about the meat science for postgraduate level. The topic is meat quality. In the previous lecture I have told there are 4 different aspects of meat quality. Now we will discuss about the physico-chemical quality of fresh meat, or sometimes we say functional quality of fresh meat. And in this today's lecture I will discuss only about pH and water holding capacity. So we will discuss what is pH and how it is important in meat, how the pH changes and what is its impact on the meat quality. And second is about water holding capacity, the chemistry of it and its impact and different kind of deviations in meat quality due to changes in water holding capacity, and how to improve water holding capacity during meat processing. So these are the different aspects in this lecture I am going to discuss, and I will have one more lecture in continuation to this on some other parameters like color, texture, tenderness, etc. Here once again let me revise the 4 different aspects I have mentioned earlier about the quality of meat or even for food. The most important are the nutritional aspect, safety or microbial quality, the sensory quality, and the functional quality or physico-chemical qualities. These are the 4 main aspects. In addition to that, there are ethical aspect, aesthetical aspect, convenience, authenticity, and origin. So all these things are also becoming important in modern times about the food and the food quality, but we are restricting the 4 aspects in quality of meat, of which the first one is the functional quality or physico-chemical quality of meat. I am discussing first part of it, and another one will be second part in the next lecture. The first parameter we are going to discuss is about the pH. As we all know, pH is defined as the negative logarithm of hydrogen ion concentration in a substrate or solution. So this is more relevant to any solution, but in case of meat, though it is not a solution, but it has got 70-75% water. So in that water the hydrogen ion is expressed and we can measure the pH. So this pH denotes the alkalinity or acidity of a solution or substance, and after that muscle pH comes down from normal physiological pH of 7.2. So we know the normal biological pH of the blood or in the muscle is around 7.2, but after slaughter, when the animal dies, slowly this pH declines and it reaches to 5.5 to 5.6, which we call ultimate pH. Now how this decline happens and how it should happen, that we are going to discuss next. We have thoroughly discussed in one lecture about the conversion of muscle to meat. There we have first discussed about the decline of pH after slaughter. So this pH changes mainly happens due to conversion of muscle glycogen to lactic acid, and then this lactic acid starts accumulating in the muscle and pH declines. So this happens due to lack of oxygen in muscle tissue. After slaughter there is no blood circulation, so there is no oxygen supply, but muscle still wants energy, and to maintain the structural integrity the ATP formation continues, and whatever glycogen available it undergo glycolysis under anaerobic condition, and that anaerobic condition produces lactic acid, and accumulation of lactic acid causes decline in pH. So in a living condition, when this happens, this lactic acid get converted to pyruvic acid in liver, but because of no oxygen, no circulatory system, no living activities, this lactic acid accumulates. So finally, as long as there is glycogen available, that continue this breakdown, anaerobic breakdown, and lactic acid accumulates, and in a normal condition this goes up to 5.5 to 5.6, which I told already, called as ultimate pH, and this ultimate pH has got important role in normal meat and meat quality, which we will see slowly. So now once again we will see the normal pH decline. A normal pH decline pattern is characterized by a gradual decrease from approximately 7.2 in living muscles to a pH of about 5.6 to 5.7 within 6 to 8 hours after slaughter, and then to an ultimate pH of around 5.4 to 5.6 within 24 hours after slaughter. So this timing varies with species. In case of large animal, the rigor mortis sets in up to 20-24 hours, whereas in small ruminants or pig it may be less time. So it is all connected to glycogen reserve and starting of rigor mortis. So this generally occurs in healthy, well-fed, rested animals. So this is the background of pH decline in a normal condition, in normal healthy animal which has got proper rest and proper practices followed. So this pH fall is achieved in the muscles of well-fed and stress-free animal. The changes, what I told from anaerobic glycolysis, it happens separately in each and every individual muscle. So the ultimate pH varies between muscles within the same carcass depending upon the glycogen reserve, the surrounding temperature, and the glycolytic enzyme activity, etc. in the muscle. So different muscle will have different glycogen reserve, and the temperature may vary between inner muscle and outer muscle, and the enzyme activity also will be different between muscles. So the conversion of glycogen and accumulation of lactic acid and pH fall or ultimate pH will be different in different muscle. The rate and extent of fall of muscle pH in the first 2 hours have considerable influence on the color, texture, water holding capacity and tenderness of meat. So these are the parameters which plays important role in fresh meat or functional properties of meat. So in this lecture we will discuss about water holding capacity, and in the next lecture we will discuss about the color, texture and tenderness of meat. Here we will see about the rate of decline of pH. So depending upon the pre-slaughter conditions of the animals and stresses during slaughter, the fall of pH can follow any of the following patterns. So there are commonly 3 different patterns can happen in the rate of decline of pH. In the left side diagram we can see there is a range of 5 to 7, and 7.2 is the normal pH in blood or muscle, as I have already mentioned. So here we can see the bottom green color, that is a very sudden fall of pH, almost reaching to 5.2 or 5.4, and in the top one little pink color, that is not falling much, very little fall of pH and remains 6.4, 6.8, quite high. So these two are abnormal, whereas the one with blue color is the normal, where it slowly pH declines and finally it reaches to around 5.6, 5.4, 5.6. That's the normal pattern. So these 3 conditions gives 3 different kind of meat. When it is normal, we can see in the right side diagram around 5.6, that's the normal meat and normal color, whereas the quick fall leading to the pale color, that is also called PAC, which I am going to explain later, and the slow fall of pH and ending with very high ultimate pH, that is leading to dark color and another condition called DFT. So all these 3 we are going to see again. So now we will see the first and common abnormality which happens more in beef, that is called dark cutting beef or dark farm dry meat. In some animals the pH fall is very negligible or very slow, only few units, and during the first hour after slaughter, and then pH remains stable at a relatively high level, resulting in an ultimate pH in the range of 6.5 to 6.8. So that's a very high ultimate pH, leading to a condition called dark, firm and dry. So this results in a poor keeping quality. Because of the high pH, such meat will have poor keeping quality. This condition occurs in animals which are physically exhausted or stressed some way during slaughter and have low glycogen reserve in the muscles. So as I mentioned, the glycogen reserve play an important role in pH decline. This we have discussed in conversion of muscle to meat in a UG lecture. So when there is continuous slow level of stress, particularly when the animals are having a long distance transport, so there is a continuous stress leading to glycolysis, and finally when it is going for slaughter, there is very poor glycogen reserve. So the normal changes, anaerobic glycolysis, do not happen after the slaughter. So that's the cause. So ultimate pH is very high, and that meat will be dark in color and firm and dry. This is because the ultimate pH is high, so it will have a high water holding capacity, which I will discuss later. And because of high water holding capacity, it looks like dry and firm, and the color also, it will have a nature of absorbing more color and giving a dark color. So here is another abnormal condition. It is due to abnormal decline of pH leading to a abnormal quality of meat, that is pale, soft and exudative meat. So animals with pH may drop very rapidly to about 5.4 to 5.5 within 1 hour or so of slaughter, while the temperature of the muscle is still above 35 degrees Celsius. Under these conditions, changes occur in the properties of the muscle proteins, and the meat becomes watery and assumes a pale, unattractive color, becomes soft in consistency and lack in flavor. So this type of meat ultimately develops a pH of about 5.2 to 5.3, even below the normal ultimate pH, and that is known as pale, soft, exudative meat. So the cause of this condition is due to acute stress just before slaughter. If there is acute stress just before slaughter, that will cause very quick glycolysis and will continue after slaughter, so leading to very quick fall of pH. And at that time the temperature is high, so that causes denaturation of protein, and due to low pH it will have low water holding capacity, so water will come out, so it will look like water is oozing out, so it is exudative. And this meat will have a property of having maximum light to be absorbed, so it will be pale in color and it will be appear like soft. So here once again we can see the decline in pH and color of meat. The topmost one, which is DFD condition, there is a very poor fall of pH and dark in color. That is because it absorbs more color, so it looks dark. Then the second one is normal, which will have a normal color, and the third one is pale, where sudden fall of pH, and that will reflect more color, so it will look light. So in the previous slide I made a mistake. This is due to more reflection of light and it looks pale. So there are three different color at three different condition or rate of decline in pH, which of course once again we'll see in the next lecture about the color. Here we can see the color changes. At the middle it is normal, at the left it is DFD, and at the right it is PAC. Similarly, in the bottom diagram we can see that pH 5.5, and when it is increasing to up to 7, the color is becoming more and more dark. So ideal point is 5.5. Here we will see the factors responsible for PAC condition in meat. I have already mentioned this is due to acute stress just before the slaughter. The important factors are like high environmental temperature. Second is rough antemortem handling just during before slaughter, fighting between animals or any other panic situation, physiological differences, and slaughtering and handling of animals before slaughter. Just before slaughter, if there is a very rough handling or during slaughter, then that will cause. So these are the factors causing the PSE. Here we will have a comparison between the PSE and DFD. In case of PSD, it occurs in a case of acute stress, whereas in case of DFD, it occurs in case of chronic stress. In PSE, initial acidification or pH decline is very rapid, whereas in DFD, acidification is very slow due to reduced level of glycogen. In PSC, the initial pH is very low when the temperature of carcass is still high, whereas in case of DFD, the ultimate pH is high. In PSC, the meat proteins denature due to very quick fall of pH and the temperature is high, whereas in DFD, meat proteins do not denature. In PSC, water holding capacity becomes low due to low pH, whereas in DFD, water holding capacity is high due to high pH. In PSC, muscle fibers almost like separate like, whereas in DFD, muscle fibers remains tightly packed, so it feels like firm. In PSC, the high scattering of light happens and that's why it looks pale, whereas in case of DFD, the low scattering of light, so it looks dark. In PSC, the surface of the meat becomes pale due to the light effect, whereas in meat surface it is dark in case of DFD. The PSC, the meat becomes soft and exudates fluid, whereas in DFD the meat is firm and looks like dry because all the water is holding tightly inside. So this is a comparison of the conditions and characteristics of PSC and DFD. Now we will discuss about water holding capacity. So earlier we have discussed about pH of fresh meat. Now we will discuss about WHC, which is a very important parameter, but it is directly related to pH. So pH and water holding capacity is directly related, and these two chemical parameters determines the important quality aspects in fresh meat and ultimately in the eating quality of meat, either after cooking or after processing. So that's why this pH and water holding capacity is a very important to understand the meat quality at every type of meat. First, here let us understand what is water holding capacity. So water holding capacity is defined as the ability of meat to retain its water against external forces such as cutting, grinding, pressing, heating, freezing. So all these external forces or mechanical forces has an influence on the meat to hold its own water. So if meat can hold the water more, that is a better water holding capacity. So this water holding capacity has a direct relation mainly with juiciness and also on color, as we have discussed earlier, and partially on the texture and tenderness. Here we will try to understand the chemistry of water holding capacity. Water in muscles remains in three forms, that is bound form, immobilized form and free form. So in the innermost part of the muscle there is bound water, which is bound to hydrophilic groups on muscle proteins and attract the water, forming a tightly bound layer of water. So in muscle there are proteins, and proteins will have carboxylic group. This carboxylic group are having hydrophilic properties. So they always form the hydrogen bonding, attract the water and make a tightly bound layer of water. This is the bound form of water. Then the next is immobilized water. So here immobilized water, here water has less orderly molecular orientation toward the charged group. That charged group, that is the carboxyl COO minus or sometime NH4 plus. So this charged group has a hydrophilic property, but in the second layer they are having less orderly molecular orientation, so they are immobilized water but not so tightly bound. And the third category is the free form of water, and they are held on by capillary forces and their orientation is independent of the charge group. So here these waters are not having such a tight holding, and that can easily come out under any minimum external forces or physical or mechanical forces. So that water goes out easily, whereas immobilized water takes little more extra effort to come out, whereas immobilized water will never come out and always remains in the muscle or in the protein. So here once again we will see the three different kind of binding of water. The first is the immobilized, that is around 4 to 5% of water is held in muscles or muscle proteins by electrically charged reactive groups of muscle protein, and they form a tightly bound layer of water in the muscle, and this water can never come out easily. It's very difficult, so that's why that is the bound form. The second is immobilized water is held by weaker bonds, and its amount depends on the amount of physical forces exerted on the muscle. So if the physical forces is more, then more of this immobilized water will go out. If the physical forces is less, a less amount of this immobilized water will be lost. And the third is free water. It is held only by capillary forces between thick and thin filaments, which we have discussed in the structure of muscle. So this water goes out very easily with minimum external forces. So here once again we will understand little more chemistry of water holding capacity. So water holding capacity reduces when it goes near the isoelectric points, that is related to protein. So in case of muscle protein, the isoelectric pH is close to 5. So when the pH goes to ultimate pH, which is normally 5.4 to 5.5, that is very close to the ultimate pH. So close to the isoelectric pH. So what is isoelectric point? This is the pH where the net charge is 0. That is the ionization of the groups, the positive ion and the negative ion, the COO minus and NH4 plus. These are the two ionization happens in the protein. When they are equal, then net charge is 0, and that happens in a particular pH, that is called isoelectric pH. Now isoelectric pH is nearer to the ultimate pH, so the water holding capacity will be minimum at this ultimate pH. But when the pH is higher, then the ionization will be different and the water holding capacity will be more. So drop in pH in postmortem period is responsible for an overall reduction of reactive groups on proteins. So pH and water holding capacity is closely related, we can see from this curve. So here once again we will understand about the isoelectric pH and water holding capacity. At isoelectric pH, a number of positively and negatively charged group is equal. Consequently, these groups tend to be attached to each other, and only those left over are available to attract water. So at isoelectric pH, the net charge is 0. So hardly any reactive group is available for holding the water. So this influence of pH on water holding capacity is called net charge effect. And higher the pH of muscle, higher is the water holding capacity. I have already told this is due to the availability of more reactive groups in muscle proteins, which help to retain more bound and immobilized water. So when the pH is high, then the reactive group changes. Number of reactive group changes, and that leads to more water holding capacity and holding more of bound and immobilized water. So overall water holding capacity becomes more. In continuation about chemistry of water holding capacity, water holding capacity of muscle is also greatly reduced due to ectomyosin formation during rigor mortis. So we have learnt earlier, during rigor mortis there is complete stiffening, and due to 100% ectomyosin bond formation, and it won't break. So this happens because of lack of space of water molecules within protein structure, which is called steric effect on water binding. So during the complete rigor mortis, ectomyosin formation happens and there is no space for water, and that leads to low water holding capacity, and that is called steric effect. Whereas in case of pre-rigor meat, it has better water holding capacity and yields higher quantity of products while converted into products. So in case of pre-rigor meat, that ectomyosin formation is not there, so it will be holding more water, and moreover the pH is high at this stage, so reactive groups are available more and water holding capacity is more. And if we use such meat, the yield will be more because of more water holding capacity, and the juiciness of such product also will be good. So sometimes it is also called as hot processing of meat. Here we can see the stages of rigor mortis and its effect on the water holding capacity. In case of There are three figures A, C and D. In A we can see there is no shortening, that is 100% pre-rigor, so the water holding capacity is very high. In the next one there is minimum shortening after post-rigor, and then there is almost a very good water holding capacity. Whereas in the further another one post-rigor, when the shortening is very high, acute stage of rigor mortis, there large amount of water is coming out, as we can see from the bubbles. So that indicates more water is coming out, so water holding capacity is less. So more the shortening or more the rigor stage, less is the water holding capacity. Once again, in continuation about impact of water holding capacity, the degree of water holding capacity has large scale effect on firmness, structure and texture of meat. Muscles with extremely high proportion of bound water are firm, have a dry or sticky texture. So this happens due to high water holding capacity, which we have discussed in case of DFD in the pH section when I was explaining. And secondly, when the pH is low. So alternately, tissues with poor water holding capacity are soft, have a loose structure and have wet or grainy texture. So this happens in case of PAC due to low pH and low water holding capacity. In continuation about impact of water holding capacity, meat with poor water holding capacity loses more moisture from its surface during storage. Water holding capacity is of special importance while meats are further processed into comminuted products, because during processing they are subjected to combinations of grinding, heating and other physical forces. So that leads to more loss of water holding capacity, and yield of manufactured product is the function of water loss from the product during processing. So if the water holding capacity is less, the more water will be lost, and finally yield of product will be less, even it will have less juiciness. Here we will see the manifestation of water holding capacity. It is manifested through exudation of fluid of meat at different stages. So when it is fresh meat and we hold it, sometimes there will be some water oozing out with a reddish color, that is called whip. Whereas when the meat is frozen and then we start it thawing, bringing it back to normal temperature, that time also a lot of water comes out with reddish color, that is called drip. And sometimes there is a loss of water due to cooking or storage of meat, that is sometimes called as shrink. So these are different manifestations related to water holding capacity. So if the water holding capacity is less, then the whip will be more, the drip will be more, or shrink will be more, or sometimes cooking loss will be more. So here once again we can see the impact of water holding capacity on the meat and its characteristics. The first one is meat with poor water holding capacity produces more drip or exudates, that is the reddish watery liquid, that weight will reduce, and the many important water soluble vitamins, minerals, proteins also will be lost, and the package will have a very bad look in the display in case of fresh meat. Then second one, the more loss of drip as well as loss of moisture from the exposed surface results in more weight loss. So that is also sometimes we call shrinkage. When meat we store it and lot of water comes out, the weight is reduced, and that happens during fresh meat storage. So that is the more weight loss leading to shrinkage. And the third is meat with low water holding capacity loses a lot of water during cooking and consequently causes lesser yield or more cooking loss, and this processed product may have a dryness and lack of juiciness. So when the water holding capacity is low, the more water will be lost during cooking, so cooking yield will be less, and the cooked meat will have a dryness and lack of juiciness. So these are the impact of water holding capacity, particularly when it is low. Here are some tips how to improve the water holding capacity, particularly for processing. In comminuted meat products, salts of strong acids such as sodium chloride are incorporated and it enhances water holding capacity of meat mix. So salts of strong acid which is added for enhancing the water holding capacity of meat. Similarly, alkaline polyphosphates such as tetrasodium pyrophosphate TSPP or sodium tripolyphosphate etc. also increase the water binding capacity of meat proteins in processed meat products. So that's why phosphate is often added while making the meat emulsion, that increases the water holding capacity, yield or juiciness. So this addition of alkaline polyphosphates results in increase in ionic strength by increasing the pH of meat mix and also by splitting of actomyosin, and thereby increases the water binding capacity of meat. So polyphosphates increase the pH and ionic strength and also splitting the actomyosin which is formed during rigor mortis. So thereby it increases the water holding capacity and it can increase the yield and juiciness. So now we are at the end of today's lecture. Today's our topic is in the meat quality or fresh meat quality, in which we are discussing the functional quality of meat or physicochemical quality of fresh meat. And today basically we have discussed details about the pH and water holding capacity. So in pH section we have discussed how the meat pH declines after slaughter and what is the normal way of decline, what happens in abnormal condition like very fast decline or very slow decline and leading to abnormal condition of PSC and DFD. And then we have seen the consequences on the meat quality, color, etc. And in the second we have understood what is water holding capacity, what is the chemistry behind it, and how the water holding capacity has an influence on the meat quality and different type of water holding capacity and their consequences. And finally we have seen how to improve the water holding capacity in processed meats. So with this I am closing today's lecture. Hope it is easy to understand and it is useful. Thank you.